Hello, everyone. Good morning, and welcome to the hybrid oral session for Alien Ecosystems, Integrating Ecology into Astrobiology. We do have a very tight schedule today. I wanted to give as much time to the speakers as possible. The speakers will have a 15-minute slot each, so in the hopes that they have about a 12-minute talk, I'll give you a 2-minute warning till the end, and then we can have some questions after each talk. So without further ado, I'm going to introduce our first speaker. We have Mario Torres Rodrigo, and you can take it away.
Good morning, Atlanta. Hello, the world. So my name is Mario Torres Rodrigo, and I'm going to be talking about today is basically my coping mechanism during the pandemic when I was just locked at home and I couldn't do any lab work. So I had this idea, and we know that in nature, minerals and microbes are intertwined. They just happen to coexist together and they impact each other. We know that microbes can affect minerals. They can dissolve them, they can modify them, and they can precipitate them. And on the other hand, also minerals are going to shape the microbial communities. So we see plenty of examples. So for example, you can take the boy out of the country, but you can take the country out of the boy. So I'm Spanish. So Rio Tinto is a massive part of my life. So we have Rio Tinto that basically is on top of Pirate. And because of the oxygen there and the presence of certain microbes, such as Acidithiobacillus ferroxidans, this Pirate is oxidized, and iron and sulfate accumulates on the water, and that's why it has this very nice red color. But also the accumulation of this sulfate and iron because of microbial activity contributes to the precipitation of certain minerals, such as jarosite. So when we look at modeling, we see that there are plenty of examples of numerically explicit microbial models that address things such as microbial growth, metabolism, metabolite production, utilization of resources. And on the other hand, we also have theoretical geochemical models that address things such as the interaction between fluids and water, and minerals, and so on and so forth. But what I found is there is this massive gap in the middle. So this is where GIMIC, which is the model I've been working with, comes to play. So the aim of GIMIC is actually numerically predict the geochemical and biological nature of brines of astrobiological interest. So how does this work? So first of all, I've written GIMIC in R, and I feed it a number of things. So one of the things I feed it with is the brine composition of the ground I want to analyze, then a microbial community, which is going to be basically 7 different metabolic groups, and then I can also add minerals to it to analyze the precipitation and the dissolution. And then what the model is going to do is calculate the concentration in the brine after the contact with the minerals using Freq-C. And also it's going to give me the activity of the different components, so then I can calculate Gibbs free energy. And finally, the water mass, in case we have things such as precipitation or freezing, so we can recalculate the things that are not included in Freq, such as, for example, the microbial biomass. So how do we model this microbial evolution over time? So we have that the microbial groups are going to be on 2 different types of pools. We are going to have active cells, and we're going to have inactive cells. Active cells are going to change over time because if they're active, they're going to be reproducing. And this is going to follow monod kinetics, and is going to also be impacted by several physical chemical parameters, such as pH, water activity, and temperature. The next thing is that cells, if they're alive, they can die, and that's a fact. So how we model death is that there's going to be a death rate that's going to happen over time. Also, we are going to have processes of activation and inactivation. We know that in the labs, we try to make conditions the best possible for microbes. And they are going to be very happy, they're going to be thriving, but in nature, we know that this is not a case. And the majority of cases, we're going to have suboptimal conditions so microbes can get into dormant states. They are still alive, but they're not going to be reproducing actively, and that's where dormancy comes to play. But what happens if the situation gets even worse? In that case, microbes go basically on a diet. They're going to be consuming their organic matter, and they're going to be producing CO2 as a consequence. And this is what we're going to call endogenous catabolism. But as I said, my main interest is trying to bring together the geochemistry and the microbiology. And we know that several microbes are going to be using different substrates and different chemicals that are going to be in the environment for their metabolisms. And we also know that for some microbes, some microbes trash is going to be some microbes treasure. So for example, if we have heterotrophic bacteria, they're going to be using this organic carbon, which is this S that I have here, which is this, the substrate, and they're going to be producing CO2, which dissolved in water is going to be bicarbonate. So that's a mineral component. So these are the different metabolic groups that I've included in the model. But yeah, this is a great idea, and whatever you want to tell me, but we have to actually show that our model works. So I decided to go to what I know, which is, again, Rio Tinto. We know that the mineralogy of Rio Tinto is basically pyrite, and the community is dominated by some microbes such as ion oxidizers and sulfate-reducing bacteria. And there's evidence that there's been anaerobic environments under Rio Tinto, which are the ones that I'm interested in. And there was this paper from last year in which they evaluated the different brines around the Rio Tinto area, and they found that the residence time for these brines is around 60 years on the subsurface. So with this information, I decided to model the interaction between the water from Rio Tinto with a shale over 60 years and see what kind of microbes we can have there. So this is what we found in terms of microbiology. So because we have the carbon leaching from the shale, this is going to be supporting an active community based on heterotrophic metabolisms, being dominated by sulfur-reducing bacteria, because Rio Tinto is super rich in sulfate, but also ion oxidizers are going to be present in our model. So if we have a bit of a snapshot of what is going on and how these different microbes are going to interact with the geochemistry, we actually find that if we analyze heterotrophic microbes, as I said, the community is dominated by SRBs, but also ion oxidizers are going to be appearing. And if we see the bottom graph, we can see that there's accumulation of substrate over time, and this is because of the leaching from the shale that I've modeled. But also we can see that there is an accumulation of bicarbonate because of the mineralization of this organic carbon due to the activity of heterotrophs. In terms of, because obviously SRBs are very important, I decided to also check what happens with the sulfur cycling. And we see that there is a small accumulation of sulfite over time as a product of the activity of SRBs, which is also going to allow the existence of sulfite oxidizers, not as abundant as SRBs, but still they're going to have an important number there. But yeah, this is great. Okay, this is great, but how is our model comparable to reality? So what I decided to do is, okay, we have this lot of brines that I said before from this paper from last year, and I want to see how similar my results are to these brines. And what I found is that if you see, this is gimmick in blue, and we have this one that is very close here. And actually, the researchers propose that this LHS Annabelle Garden actually comes from the interaction of Rio Tinto waters with shale. Aha, which is exactly what I modeled. So literally, I screamed at my computer when I saw this. And what happens with the microbiology if we compare with Rio Tinto? So Rio Tinto has these two kinds of environments. Some of them are dominated by ion oxidizers, such as Acidiphia bacillus. But then there are other environments that are basically hugely dominated by SRBs, such as Syntrophobacter. And Syntrophobacter is a microbe that reduces sulfate utilizing organic matter, exactly what I model on Gimmick as well. So just wrapping up, Gimmick integrates geochemical and microbiological data, which is my aim, so that's good. The results from Gimmick reproduce accurately observations in Rio Tinto. Gimmick can be used to predict challenging ecosystems such as subsurface environments because they are very hard to drill and to get aseptic samples from there because you are very exposed to contamination. And also, I've used Rio Tinto because apart from knowing it, it's also a very good astrobiological analog for Mars in the past. So actually, the next step maybe is actually using Gimmick on Martian brines or on icy moon brines, but also I want to explore other avenues and model other environments on Earth, such as freezing brines as we have in blood flows, which actually what I was going to originally present here, but the results were not quite ready yet. So this is why we finally moved to Rio Tinto. And that's it. That's from me. So if you have any questions, more than happy to take them.
Hi, Tessa Fisher, ASU. Great talk. Once upon a time, I was a microbial ecologist modeler. And the first thing that came to mind, aside from the fact that I would have loved to have this back when I was working on my projects, how did you factor in sort of like the fundamental nutrients like, say, phosphorus availability?
So in terms of phosphorus and such, Frick actually can model, well, Frick doesn't really model phosphorus. So we've actually added some of the picture data into the database to try to model it. But also in terms of microbiology, one of the things that we are modeling is actually the incorporation of phosphorus into the microbial cells. So we know that there is an affinity constant for phosphates for microbial groups at the time of incorporating it. So basically, the dynamic is, okay, if there is organic phosphorus on the brine, it's going to be absorbed by heterotrophs to generate organic matter. And if we have inorganic phosphorus, it can be also absorbed by autotrophs to generate the inorganic matter. On the other hand, if we have organic matter, it contains nitrogen, it contains phosphorus, and it contains also carbon. So when we are actually modeling processes such as the mineralization that we see by heterotrophs, we are actually also putting the phosphorus dynamics into it because we know the percentage of the organic matter that contains phosphorus. Okay. So that is also being mobilized into the brine.
Makes sense. Thanks.
Thank you very much. Any more questions? Okay, a round of applause for our first speaker, please. Okay, next up, we're doing great on time. We have Laura Fackrell. So please give a round of applause for our second speaker.
Working. I can't see the thing, so I guess it's working. Okay, perfect. All right. Good morning. I am Laura Fackrell, and I am a postdoctoral scholar at Northern Arizona University. And today I'll be talking about a project we're working on where we're developing habitat suitability models and different species distribution models for some taxa in some of the polar deserts and how we're applying that to Mars. So before I get into the details of the talk, just some acknowledgments from my fellow co-authors, as well as other collaborators and students that have worked with us on the project and several funding sources that have supported it. Okay, so I'm going to give you a little bit of context first, like where exactly, the project is coming from. And then I'm going to talk about some of the methods and preliminary results from like the first steps of the project. We are still kind of at the beginning of this project. It's an ongoing thing, and so we don't have any actual habitat suitability models finalized yet to show off, but we do have some early results that are pretty cool that I'm going to talk, the methods and the results I'm going to talk about, we'll focus on that today. That'll focus mostly on soil moisture. And then we'll kind of talk about what's happening next and where that's going. All right, so first some context of where this is coming from. This is actually a project, the main project that this is coming from is a project we're working on in McMurdo Dry Valleys in Antarctica, and it's called Moving Beyond the Margins. And we're trying to model water availability and as well as different habitat suitability aspects of these polar deserts and some of the major goals that relate to specifically water and other aspects of the project. And these are not all of the goals of this pretty large project, but it's the goals that are most applicable to what we're talking about in relation to then relating that to Mars environments today. And that is mapping the sources and distribution and abundance of soil moisture in these soils, especially cryptic sources that are not necessarily accounted for as well as some others, and we'll talk about that in a couple of slides. We're also going to look at the next steps after that are assembling other data sets that are geospatially relevant and relevant to the taxa that live there, and that would contribute to understanding the habitat suitability of these regions. And then taking all of this and developing species distribution models and other related kind of models of that to kind of understand better the species distribution of these different areas and how these more cryptic water sources may be contributing to that, and as well as how climate change might be affecting that. So that's kind of like the bigger project overall, where this smaller project is coming from. Because these McMurdo Dry valleys are a Mars analog, we've also wanted to see what information is available and what data is available, so we can take this approach that we're using in an Earth ecosystem environment and then apply it to a potential ecosystem on another planet, specifically Mars. And so why particularly soil moisture, and why that's kind of the focus of what we're talking about today? Well, it's kind of like that first big goal of what we're talking about. And as astrobiologists, we know that water is really important for life. And so like following the water and understanding how the water behaves is a really important aspect of any environment. And in particular, these polar deserts where water is extremely limited, some of the driest places on Earth, so dry that they actually are some of the best analogs we have for Mars because of just this nature of these deserts. Then we can actually kind of look at what are the behaviors of water in this valley. And we have this kind of classic view of McMurdo Dry valleys where you have seasonal and glacial snow melt that are feeding these ephemeral streams, and that they are the main contributor to different processes that would involve ecosystem processes as they depend highly on this water. And that is a major part of what happens in these ecosystems. However, there is a recent challenge to this paradigm where we're actually starting to find other sources for water that maybe have a greater contribution than we before realized. In particular, the detection of several wet patches that would be called the wet tracks, that are these shallow subsurface systems that are contributing water to the environment, but they aren't part of this major stream flow. And so we've been able to actually kind of not only estimate the abundance and the distribution of water from streams, but also from these cryptic sources that are not really very well accounted for and are kind of only recently being more studied and understood, and could have really potential importance in how ecosystem processes function in these environments, and have a lot of potential for helping us understand extraterrestrial ecosystems if and when we find them. So that's kind of, but they're also very different from the streams themselves because they're often more highly saline. So this really interesting kind of approach trying to understand the soil moisture. And that kind of takes us to recurring slope lineae, or how we want to apply that to this particular environment. This is not the only environment that this could be applied to, but just kind of one that we picked because there's a lot of correlations and comparisons that could be made from these wet tracks that are in Antarctica to recurring slope lineae, but there are also some important distinctions and clarifications to remember for them. And so we know that recurring slope lineae are a pretty familiar thing on Mars now, are these dark linear streaks that appear yearly along slopes and on steep Martian slopes. And then they kind of tend to be seasonal, where they appear in the warmer summer and then kind of disappear in colder temperatures. And initial observations of these features, and even some detections or preliminary detections of salts within them, kind of supported the initial thoughts of them being these wet mechanisms that supported them. However, a lot of more recent observations have kind of clarified that, and we've kind of gone away from the wet mechanisms and more towards dry mechanisms, just because the salts that were detected were not necessarily robust detections, and so that's been highly disputed. But there's a lot of different opinions in the literature and different things, and none of the mechanisms fully support all the features seen, and so there's a lot of debate still. And there's still a lot of debate on whether or not they're wet or dry. But that's not what this talk is about. We're not trying to figure out if they're wet or dry. That's a bit beyond the scope of what we're doing. We need more data to really figure that out anyway. But it's if they're wet, or if there's any kind of wetness involved, how wet are they, and what can we say about that? And that's using some of the same methods that we're applying to Antarctica, trying to apply them to these methods, where this wetness is an albedo feature that can be observed and measured and potentially even pulled out from the rest of the data. So that's where and then also just to clarify really quickly, habitat suitability versus habitability. And so when I'm talking in this talk, I'm talking more about habitat suitability, which is a pretty strict ecological term on which a lot of these habitat suitability models and distribution models are based. And it's a little bit different than just habitability that we often talk about in astrobiology. So it's just a quick clarification to make about that. All right, so how are we approaching this, and what are we doing to do this, the soil moisture aspects of this? And then a little tiny bit about what's coming to get to the actual models in the end. Okay, so first, this is just an image of some of the lakes that are in these McMurdo Dry valleys and the changes that occur there. And you can kind of just see how dynamic it is and how much the surface changes. And a lot of those changes are just these albedo differences that you can see in the background, where you just have this change from light to dark and different things. However, a lot of things can influence albedo, from composition to topography, and a lot of other features that just play into that role. And so how do you isolate the albedo changes that are from soil moisture specifically? Well, we're fortunate, we're in an area that has no vegetation and no animal life. So we just have this open canvas that we can kind of look at. And we also have really detailed DEM and topography data from LIDARs in this area as well. So we can kind of isolate the features that are from topography and then separate them out. And so that's kind of the approach that we used. So we took these high quality image data from Worldview 2 and 3. And we have about 57 images total from all of that from the years 2009, 2019. We calibrated them and corrected them atmospherically and all the surface reflectance and all of the different things. And then we were able to use the LIDAR data to construct a really accurate hillshade and other topography relevance that we'll be able to model the topography of the area. And then you can use that to then subtract that out. And then we get to that third image on the bottom, and that's what you remain. So you remain with the albedo changes that aren't influenced from topography. And so that leaves behind what you can then use to calculate, okay, what are the other things that would influence albedo in this area? And because the composition is relatively consistent, and because we understand the relationship of different salts and snow to this area, we can kind of isolate what is from potentially wet patches if we look at it over time. And that can kind of isolate that those changes in albedo that occur over time in the seasonal patterns can then be from these wet patches, as well as streams and other sources of soil moisture. And we've actually also correlated this to studies in the lab, where we've been able to have, there's a fairly linear relationship before the soil becomes saturated. So once you get saturated, it gets a little more complicated of spectral relationship, but before that point, you actually have this really nice linear relationship between the soil albedo changes and the soil moisture content. So we're able to use that to actually then help to also estimate how much is there. And because we have data from the ground in Antarctica, we can actually compare that and show that it is similar. And so we've been able to do that. So that's kind of the soil moisture aspect of it. And then really briefly, we're also integrating it with other meteorological data, different NDVI index, all sorts of other geospatially related data that helps us to understand the habitat suitability of this region and incorporate into these species distribution models. And that's, the lower example is just an example of wind distribution in the area. All right, so then how do we apply that to RSL? Specifically just focusing on the moisture, soil moisture aspects, if it is indeed moist at all. And that's using, and so we carry over from Worldview to high-res images, and we do similar corrections. And also we have enough high-res images that we can produce a DEM of the area that's accurate. It's not quite as detailed as what you would get in Antarctica, where you have this nice LiDAR, but it's accurate enough that we can kind of do the similar workflow and then get an estimate for the soil. If these albedo changes are indeed from soil moisture, how moist is it? And then we take those images and just put them through a very similar workflow as we did for the Worldview images and apply them to these RSLs. And then that will be assembled with other available data that's, as long as that data has a proper resolution and is the kind of data that we need to that we can combine that and perform a more limited kind of habitat suitability model. We don't have as much data to work with, so we can't do as many things as we could do in Antarctica, but we can start to understand what are the contributions to habitat suitability from water at least, and from other features that we actually have available data for. And so just kind of take this example and apply something that we use all the time in ecosystems on Earth and see what we can do with it in an environment on another planet. So that's kind of the approach there. Okay, so just a couple of preliminary results. So this is just what we would see in Antarctica. So kind of just a really quick visual is we do want to look at the variation of albedo over time. If it's just a stagnant albedo that doesn't change at all, then that can likely be due to composition or other features that affect albedo. But if you can look at the ones that change seasonally that match this pattern, we can actually kind of understand that variation from the average and get an idea of soil moisture. And so applying that same thing to Mars, this is kind of the result for Pellicer crater, which is the first one we tried this on. And we're also going to try it on a Hurwitz crater kind of in the next phase because we have enough data there, enough images to actually do this. And so that's kind of, this is that result of like the estimate of volumetric water content if that albedo change is from water. And so that kind of got us this result where we have around 10 to 20% in RSLs with less than 5% if it's not in RSL. So we have like this range of what the soil moisture content would be. None of them have been above 20%, so it's not a lot of water. That's not saturated. But it is within a comparable range for what we saw for the Antarctic soils. And combined with some of the other data we have in the thermal modeling and different things, it's actually comparable to some other results that people have seen using Themis to try to estimate moisture abundance. If you can take into account Themis's resolution and the fact that Themis is a nighttime measurement, then it's within a comparable range that's consistent with what other results have found. And so then we want to take that and combine it with that other data to kind of get more of a habitat suitability approach. So if you're interested in that, we don't have those results to show yet, but they are coming. And so if you're interested in that, just talk to me afterwards and we can kind of keep in contact and update when that happens. But for now, we're just really excited that we have just this idea of like, if they are wet, then we can understand how wet they might be and what implications that might have for habitability. And so I'll just kind of leave up our conclusions and take any questions. Thanks.
Okay, we have plenty of time for questions, so.
Hi, Evo Mendez from PHL at UPR Arecibo. Nice talk.
Thank you.
And I am glad that you're using habitat suitability instead of the general term habitability. And we would like to help you because we are working on those models and trying to help other people incorporate a more standardized models for habitability. Mm-hmm. That was my comment. Thank you very much.
Thank you.
Hello, great talk. I'm Garrett Roberts Kingman from Ames Research Center. This is a little bit tangential to what you talked about, but I was curious, because it's relevant to the habitats for these organisms. Do you have estimates on the salinity of these RSLs and how that may differ between your Antarctic modeling and ours?
And that's a great question because that's a big part of it. And that's probably a question that was more answered, but has become less answered as we realized the original salt detections that they had for these RSLs are actually not robust. And so we don't know if there are salts in them or not technically.
Okay, thank you.
I'm going to piggyback off of that question. So I saw a talk by Jacob Schaefer here measuring the soil conductivity of the wet tracks in the Antarctic Valley, showing that compared to adjacent dry patches, they have something like 1,000, 5,000 times the conductivity. So with your results, would you expect something similar on Mars, or?
I think if it is a similar mechanism to the wet tracks, then yeah, I think we would expect something like that, depending on what the actual salts would be. But there's a lot that we don't know about RSLs yet, so there's a lot of assumptions that we're making. But yeah, the wet tracks are very much more saline than the freshwater streams that are typically in these areas. So it's a very different kind of wet environment than we would think of typically.
Yeah, of course. And
yeah.
I have a sort of secondary question to that. The work in Antarctica shows that a lot of those wet tracks can be influenced from the permafrost below.
Yeah.
Do you think that there's a similar mechanism on Mars, or?
There could be possibly, but there's and permafrost is definitely one of the mechanisms for the wet tracks, but there's also deliquescence could also be a mechanism, and there's different mechanisms depending on which wet track and like how saline, like there's a lot of different places, but that could be potentially one source for moisture in a Mars environment, because we don't necessarily, especially from the more recent observations, we realize they're not, if they are wet, they're not very wet. RSLs probably aren't being these large brine flows necessarily. Like what is the source for those brine flows? But a permafrost or deliquescence maybe, depending on how you look at it. But again, we don't really understand how much salt there actually is. And so there's a lot more data that we actually need to understand for. But I wish we could just take a rover and stick it in an EC meter and just like measure it and just like.
Yeah, we need good mineralogy of those, RSL. Yeah, thank you.
Yeah.
Do you have any more questions? Awesome.
All right.
Thank you.
Thanks.
All right, next up we have Hart Bather. So please welcome Hart to the stage.
Sounds good.
Cool. Hey, everyone. My name is Hart Bather. I'm at CU Boulder working with Sebastian Kopf and Alexis Templeton. My current project is calibrating the hydrogen isotope biosignature of archaeolipids in a model methanogen. Cool. So life forms a bunch of different macromolecules that can be used as potential biosignatures, so like proteins, lipids, carbohydrates, and DNA and RNA. Proteins, carbohydrates, and DNA and RNA are really chemically fragile and unstable over long periods of geologic time. However, lipids are pretty stable and can persist up to like hundreds of millions of years over geologic time. So that's a really important molecule to look as a potential biosignature. Cool, so like what even are lipids? Lipids are nonpolar hydrocarbons in their most reduced form, which means they're super great for like energy storage, because once they're oxidized, they release a bunch of energy that could be used by the cell. And they form things such as waxes, oils, and cell membranes. So this is a cell membrane, and you can see on the bottom is an individual lipid. It's made up of a polar hydrophilic head and a nonpolar hydrophobic tail, which is important to keep in mind when I go over my methods. And so this is basically all the domains of life with their specific lipids that characterize them. And I'm just going to go over each one and basically what lipids are used for each of them. So lipids can be used to basically determine different like environmental and physiological factors regarding that organism. So we're going to start with eukaryotes. So eukaryotes are pretty well studied, and in eukaryotes, specifically like plant waxes and other photoautotrophs, the D2H or hydrogen isotopes of the lipids can tell you about past hydrological cycles. So this is an image that shows you on the y-axis the D2H of different plant waxes, and on the y-axis you can see, x-axis you can see rainwater, and what you see here is a pretty positive correlation. So basically by looking at the D2H of these plant waxes, you could determine past like precipitation patterns and hydrological cycles. Bacteria are also super well studied as a domain, and their D2H tells you about their metabolism. So this figure shows you that there's different sources of hydrogen for bacterial lipids. So you have water, you have NADPH, which is a hydride carrier in the cell, and you have organic substrates for heterotrophs. And then on the bottom part of this image, you could see that on the y-axis, you have the lipid deuterium content, and on the x-axis are different metabolic pathways. So you have like autotrophic pathways and heterotrophic pathways. And what I want you to get from this is you could have different hydrogen composition of lipids based on your metabolic pathway. And the D2H ranges over like hundreds of per mil, which is large even in hydrogen space. So archaea are the last domain. They're not really well studied in terms of isotopic biosignatures. Archaea are kind of like bacteria, but they have slightly different cell structures and they live in extreme environments. And although they're able to live in these like super extreme environments, they don't do well in like really comfortable labs where they're given everything they need. They just choose like not to grow and stuff. Why? I don't know. But basically, they're still really important to study, even though there's been a lot of like lab and analytical techniques. They're important to study because they do live in these environments and are important for like nutrient and energy transformation in really chronically nutrient limited environments that are analogs for those like found on other planets. Methanogens specifically are a type of anaerobic archaea that produce methane, and they live in environments that also that are also very limited and mimic those found on like Mars, Europa, Enceladus, etc. Methanogens specifically are a primitive metabolism that are thought to be one of the first forms of energy transduction. They contributed to early Earth's reducing environment, and they're thought to be super important in the origin and evolution of life on Earth. So here we have two images. One of these extreme environments that methanogens live in are serpentinizing systems. So I'm sure you guys have heard a lot about serpentinizing systems at this conference. The top image is Oman, the bottom one is Mars. As you can see, they look super similar. Serpentinizing systems, so Oman is a low temperature serpentinizing system, which basically means that there's water rock reactions in this system that produce a bunch of hydrogen and also makes it really carbon limited and produces alkaline to hyperalkaline waters. And Oman and other serpentinizing systems are thought to are analogs for those found on other planetary bodies. So it's really important to study methanogens because, once again, primitive metabolism, and also they're present in environments that are analogs for other bodies that are important for astrobiological exploration. So yeah, methanogen lipids are an untapped source of modern and ancient environmental information. So that's my main point, and my research questions are: what processes determine lipid hydrogen isotope signatures in methanogens? How are hydrogen biosignatures of methanogens influenced by physical and chemical environmental parameters? And do methanogens demonstrate physiological adaptations to carbon limitations in these serpentinizing systems? My overall goal is to create a framework to understand information stored in archaeolipid hydrogen isotope ratios. Methanogens are grouped into three main groups depending on their substrate usage. First, we have hydrogenotrophic methanogens, which basically use H2CO2, formate, or a couple simple alcohols to reduce CO2 to methane. Next, we have acetoclastic methanogenesis, which breaks apart acetate to form methane and CO2. Lastly, we have methyllotrophic methanogenesis, which uses methylated substrates such as methanol and trimethylamine to form methane. I'm going to be focused on hydrogenotrophic methanogenesis and acetoclastic methanogenesis because these are the most common ones found in nature. So I'm going to do this by looking at three different species of methanogens. First, we have Embarkeri, which is a super cool bug because it can participate in all three pathways. So it's hydrogenotrophic, acetoclastic, and methylotrophic. It's well studied, so it has a simple, fully sequenced genome, a bunch of lipid information, and it's environmentally diverse, and that it can be found in like fresh water as well as landfills and like cow guts and stuff. Next, we have Ember Pollutus, which is a hydrogenotrophic methanogen that could also use formate as both energy and a carbon source. It is also well studied with a fully sequenced genome and is found in marine environments such as salt marshes. Lastly, we have Methanobacterium Nshq4. So this is a hydrogenotrophic and fermatotrophic methanogen. This is my exploratory system. So this is the one found in those serpentinizing systems in Oman. Not well studied at all, but I'm using Ember Pollutus as a model system for this bug. And yeah, so it's found in the serpentinizing system, which is the Samail ophiolite in Oman. And so this is like the schematic of what I plan to do. So you have the super primitive metabolism, methanogenesis, where you have the production of methane as well as biomass. But like what makes up the lipid hydrogen isotope signal? Is it like water? Is it hydride carriers, acetyl-CoA, which is an important precursor in the formation of biomass? I aim to do this with two different methods. So aim 1 is focused on batch cultures, aim 2 on chemostats. So aim 1, I hope to test the impact of carbon source and energy availability on lipid hydrogen isotopic composition, as well as lipid production. So for this one, I'm going to use all three methanogens, grow them in like hydrogenotrophically, fermatotrophically, and acetoclastically by using three different heavy waters. So +0 just means molecule water, and then enriched by 225 per mil and 450 per mil. For the hydrogenotrophic, since they use H2CO2, I'm going to pressurize the headspace with H2CO2. For the other substrates like formate and acetate, I'm just going to purge with N2 because I just need to keep it anaerobic or else they will die. And so basically, I grow these up in batch culture experiments in these continuous OD readers up until they reach stationary phase, which looks like this, like growth curves. My next aim is to test the impact of carbon availability and energy flux on the hydrogen stable isotope fractionation of the produced lipids. So for my batch cultures, are going to be grown in like ideal conditions, like the temperatures they like, the pHs they like, excess everything, so they're happy. I just want them to grow. For this one, I'm focusing on kind of testing parameters out. So I'm using chemostat, which is a continuous culture. Basically, you have like an influx of media that equals the outflow of media. And this is great because you can keep the system in steady state by changing and keep the system in steady state, but you could also change parameters to see their impact on the cultures and then sample it immediately to do analyses. So for this, I'm using the hydrogenotrophic pathways of Ember Pollutus and my exploratory system, the Methanobacterium, just because of time constraints. Chemostats are super finicky, don't work a lot of the time. So I'm trying to keep my chemostat work feasible. I'm going to test 2 carbon availability conditions, so excess carbon, limiting carbon, 2 energy states, excess H2 and limiting H2, and 2 pHs, neutral and alkaline. Alkaline because that's the waters that the Methanobacterium are found in. Cool, so once I have my cultures, I'm gonna do a couple different analyses. First, I'm gonna measure the headspace. So basically just like to take a sample of the gas, insert it into a GCFID TCD to see the concentrations of methane, CO2, and H2. And then I'm gonna take the cultures and harvest the biomass. And then I'm gonna do a lipid extraction, and this is like a 2-step protocol. The first step breaks off that head group, which is that polar head group, if you guys remember that image I showed you earlier, and this isolates the alcohol. After that, I'm going to cleave the ether bonds in order to produce phytane. And the phytane, which is that last molecule down there, is something that I could run on additional instruments. Then I'm going to run these lipid extracts on a GCFID first, and that's for quantification. And so basically, you just get some peaks. One is going to be your analyte peak, analytes peak, and then the other ones are just going to be standard, so you just compare to see how much you have. Second, I'm going to run it on a GCMS for classification, and so this, you get a bunch of peaks, you pick one, and then you look at the fragments that the compounds are broken up into and compare those of your target analytes. Lastly, I'm going to run on a GCRMS to do hydrogen isotope fractionation. So I'm like supposed to have data on this, but our GCRMS broke a couple weeks ago. So I've been working on that. Not successful. And also we have a helium shortage, in case you guys are wondering. So yeah, eventually I'll have data, hopefully. But yeah, so because I don't have data, here are some predicted trends. So on the y-axis, we have rates of growth and methane production. X-axis are different parameters. So with pH, I hypothesize that we're going to have the greatest growth and methanogenesis at ideal pHs. So that's like neutral for Ember Pollutus and Barkrai and slightly alkaline for Methanobacterium. In terms of carbon availability, if we have limiting carbon, you're not going to get as much growth or methane. And once you have saturated, greatest growth in methane, and once you reach excess, it kind of plateaus out. That's similar for energy availability or like hydrogen. And then lastly, carbon source. So I anticipate we're going to have different rates based on the different substrates that we use, acetate formate H2CO2, just because they have different Gibbs free energies, so different free energies available to actually do these metabolisms. In terms of lipids, I anticipate that there's going to be different contributing factors to the lipid D2H, so intracellular water, and then hydride carriers, as well as acetyl-CoA, which is important, once again, precursor for producing biomass, and that will produce these different classes of isoprenoidal ether-bonded lipids. In addition, I expect carbon availability, energy availability, and pH to have an impact on the different classes of lipids made. So in certain like nutrient energy limited environments, the methanogens may produce other specific classes of lipids over others. Lastly, in comparison to how like carbon fractionation is impacted by nutrient and energy availabilities due to enzymatic reactions, I anticipate that they're also going to have an impact on the hydrogen isotope signals of these lipids as well. So yeah, this is a study looking at slow growth and metabolism, more commonly found in natural settings, insight into information stored in archaeolipids, and a framework to assist in future inquiries into primordial metabolisms and our ability to detect life on other rocky bodies in the universe. And I'd like to thank my advisors, Alexis Templeton and Sebastian Kopf, members of T-Lab, especially Eric Ellison, members of Kopf Lab, Adam and Jamie, and NSF for funding, and the Mars Exploration Program at JPL for funding my trip out here. But yeah, that's it.
We'll take any questions.
Hi, that was a great talk. I really, really enjoyed it. And I have a question. I'm a sucker methylotrophic methanogenesis. Are you going to try Bakary also with trimethylamine or DMX of the sources to see how the lipids change as well or not?
That's a good question. I currently do not plan to, because that's kind of just not what is found in nature most of the time. For the bark rye, I wanted to do hydrogenotrophic because the Ember Pollutus and Methanobacterium are also hydrogenotrophic. So I want to see if it's like, if the actual like different methanogens have an impact on the D2H of the lipids. And then I was going to do acetoclastic as just a comparison because that's also found in nature. I wasn't going to do any of the methylated substrates because of like time constraints, but it's definitely something to consider, just not my priority right now.
You know, also one of the things I was thinking is, especially in very saline environments, if you have sulfates, especially,
Yeah.
methylotrophic methanogenesis is the one that takes over the rest of the methanogenic routes.
Yeah.
So that's what I had in the back of my mind. But I perfectly understand the limitation of time, especially with the heliums per day.
Yeah, exactly.
Well, thank you.
Yeah, thanks.
Fortunately, we have time for just one more question.
Thank you. I'm Catherine Wright, formerly in the temperature lab at CU Boulder. I graduated some time ago. Really great talk. Thank you. I'm just interested in your choice of hydrogen fractionation rather than carbon. Is it just that it's been less well studied, which would be a great reason, or is there some other reason as well?
Oh, yeah, great question. Should have mentioned that. So hydrogen isotope fractionation in archaeolipids is not studied very well. There's like one paper, Wu et al. 2020, and they kind of focused on hydrogen comparing to carbon fractionation in lipids. And because hydrogen fraction, like the hydrogen composition of like eukaryotic and bacterial lipids can tell us so much information about like past environments or like physiological adaptations of these bugs. And because archaea are super important to study in terms of like looking at primordial metabolisms and biosignatures on other planetary bodies, I thought that like studying on like hydrogen isotopes in archaeolipids can give us like really important information about the past. But I also didn't put on here because I didn't want to go into it because it's like a 10-minute talk. But I'm going to do carbon isotopes of lipids as well as a comparison. And as well as like I'm going to look at the isotope fractionation of the methane produced.
Thank you.
Yeah.
Thank you very much, Hart Bather. Next up, we have Rachel Moore.
Oh, thanks. Okay. Hi, everyone. So my name is Rachel Moore, and today I'm going to be talking to you about something that I also started during the pandemic, similar to what Mario, our first speaker, was talking about. So I was excited to hear I'm not the only one. And I'm a postdoc here at Georgia Tech in the Planetary Exploration Lab, and today I will be talking to you about genome-scale metabolic modeling as a tool and how we have used it to assess the habitability or the habitable, the potential and production of biomarkers in an early Mars Paleolake at Gale Crater. So pictured in the bottom right-hand corner of this screen is a visual representation of a simple genome-scale metabolic model. We have an organism whose genome has been fully sequenced. Those genes have been linked to proteins, and those proteins were linked to reactions. And since we have all of the reactions available, we can then model the flow of metabolites through that model. That is, we know where certain compounds can be consumed and where they will be produced, and we can then constrain this model by giving it certain inputs, like a defined media, and we can compute the outputs of metabolites and biomass production rates. So to go through this, I'm first going to talk a little bit more about genome-scale metabolic models, that process, and give some background there. Then I'm going to show some preliminary results from the model that we did of a lake at Gale Crater. And then I will talk a little bit about predicting biomarker concentrations. So like I said on the first slide, to build a genome-scale metabolic model, we first start with an organism whose genome has been fully sequenced. Those genes from the annotated genome are then linked to reactions. Next, all of those reactions are integrated through their shared metabolites, so this results in a construction of a metabolic network for that organism of interest. Then the metabolic network can be converted into a stoichiometric matrix, or S matrix for short, where the rows represent metabolites and the columns represent reactions. Each entry in this matrix represents a reaction coefficient of a particular metabolite in that reaction. So zeros indicate that that metabolite's not present. A negative number means that it's being consumed in that reaction. A positive number means it's being produced. This process is now fairly automated. We can use online tools like K-Base, but you still have to go through the literature and make sure that you are only putting in things that actually belong in that organism and you're not creating something new unless, of course, you're in bioengineering. Once this model is solved, we can then identify key features of metabolism like growth yield, resource distribution, gene essentiality. And to actually solve this model, we use linear programming through flux balance analysis to find the optimal solution for a single reaction. And this reaction is one that the organism should theoretically optimize or could theoretically optimize, like biomass production, for example. And we also, of course, need to constrain this model by providing it with a defined medium. So we have used this framework to model a very simple community and a very simple representation of Gale Crater. But before I talk about the organisms that we chose and why, I wanted to mention that these are by far not the only life as we know it that maybe could have survived in this environment. In fact, there are quite a few people that have written about the plausible microbial metabolisms for life on Mars, and this is because there are quite a few different metabolic species present there. And maybe some of these people you've actually heard speak this week. But we have to start somewhere, right? So in our case, we started by thinking about this previously published experiment as these two microorganisms could feasibly grow in the early Mars environment. Rhodopseudomonas palustris is a bacterium that is capable of 4 different modes of metabolism, or using 4 different modes of metabolism. And one of those, it can grow photoautotrophically using sunlight without oxygen. Geobacter sulfur-reducens is an obligately anaerobic bacterium, meaning it will die with oxygen present, and it is primarily heterotrophic. Both of these organisms are really interesting because they're capable of utilizing solid-phase iron oxides for extracellular electron transfer, so they can use things like magnetite. So in this co-culture experiment that you see on the left, this group, Byrne et al. In 2015, show that R palustris can oxidize iron 2+ in magnetite using light energy, and G sulfur-reducens can reverse that by reducing the Fe3 that was made, turning it back into Fe2. So essentially, they're using the solid phase magnetite like a rechargeable battery, and that's why these organisms were of interest to us. So these known Earth organisms are the basis for our preliminary model of Gale Lake. Coincidentally, both of these organisms have already had metabolic models created for them, and they have been validated against growth data in typical laboratory conditions. So we have models that are accurate at predicting the growth of these organisms in typical laboratory media, not astrobiological conditions. But that brings me to our next step. How do we actually define the media based on real data, in our case from Gale Crater? So we all know, of course, Curiosity has been analyzing the chemical compositions of sediments at Gale Crater. More recently, groups like Fukushi et al. In 2019 have taken the chemical compositions of and the presence of certain minerals and used it as constraints for thermodynamic modeling using React from Geochemist Workbench to reconstruct the water chemistry at Gale. So this is one of the tables from their paper. You can see that they took data from John Klein and Cumberland drill sites and inferred chemical concentrations for that lake water. So we have utilized this information along with other data sets to define our in silico media. So following the methods of Marinos et al. In 2020, this is just an example. This is not the full in silico media that we're using, but what we did was we mapped the chemical compounds from these data sets to metabolite identifiers of the metabolic model, and then set the upper bound flux to those concentrations. So this allows you then to get an estimate of the maximum potential biomass produced from these specific metabolisms, if anything grows at all, of course. So again, this is just an example image of an in silico defined media, and we set this all up using CobraPy and Python and solve these models using the Gurobi solver. So just to recap so far, our simple model, we have 2 organisms that we've identified as being feasible in this environment. Again, just a starting point. We're in this liquid water environment. We also know we have certain trace nutrients and metals coming from that data we have from the sediments. We also probably have some amount of hydrogen that could be coming from a variety of sources, maybe radiolysis, maybe serpentinization. We also have probably a thicker atmosphere. We're in the late Noachian or early Hesperian period, and from that we know there was probably some amount of nitrogen, carbon dioxide that can diffuse into this system, and finally, since we're in a lake system, we also have a photon flux. So if these organisms are able to grow in this environment, they should be able to cycle this iron like we saw in that co-culture experiment. These are just some of these citations. So that's exactly what we saw. So first we did see that the microbes grew individually in the given lake media without living in a community. So they did grow. They were capable of surviving with only CO2 as a carbon source, but we found that they grew at a much faster rate when combined in the same environment, and this indicated syntrophy or cross-feeding. And we did in fact see the microbes cross-feeding. Both actually produced carbon compounds and exported it, and the other organism was capable of taking it in and utilizing that. And these values are just the fluxes of these carbon compounds being exported or imported into the cell. And then we also found that iron was effectively cycled as well between redox states. So G-sulfur-reducens was reducing iron 3 to produce iron 2, and then R+tris was taking that and turning it back again. So they did this at identical rates, hence the cycling. And again, these oxidation states of iron can be found within magnetite. So next, what I wanted to do was determine the maximum cell concentration that could actually be maintained within Gale Lake. And this is because I'm a microbiologist by trade, and I want to be able to compare cell concentration to modern day analogs of the environment. So to do this, we used a dynamic flux balance analysis, which is similar to the traditional flux balance analysis to solve this community model. And a dynamic flux balance analysis, it simply couples a dynamic system to, in the external cellular environment to the pseudo steady state of the metabolic model. So we basically ran this model for individual time steps and allowed the flux of metabolites in the media to be consumed sequentially. So from there, then we were able to predict how much biomass could grow per amount of nitrogen, which we found to be a limiting growth factor in our case. And we ran this dynamic analysis using both a low and high bounds of media flux based on the findings at Gale Crater. So these media bounds were defined from the high and low ranges of chemical compounds or chemical concentrations from that Fukushi paper. So starting with 0.1 grams of biomass, you can see this in the solid line, we grew about 0.23 grams of biomass per gram of nitrogen. This is regardless of media flux. So using this information, the amount of biomass grown per gram of N2, the weight of a typical cell, the amount of N2 that could possibly be in this environment, we can then calculate an upper bound concentration of microbial cells. And depending on the concentration of nitrogen present, Gale Lake could have potentially supported, again, this is a model, as much as 10 to the 5th cells per mil of lake water. So again, this calculation is just based on the amount of biomass produced and assuming that cells are a picogram in weight. So this is incredibly similar to what we see here on Earth in a number of oligotrophic water-based environments. For example, eutrophic brines and solar salt harvesting facilities, you can find anywhere from 10 to the 5th to 10 to the 7th cells per mil. And subglacial lakes, on the other hand, we can find anything from like 10 to the 4th to 10 to the 5th cells per mil. So this is very promising that we're finding similar overall concentrations. But again, this is assuming that everything is well mixed and distributed throughout the depths of Gale Lake, and this may or may not have been the case in reality, but could be a good starting point. So finally, I mentioned on the first slide that I will also be talking about biomarkers, and this is why I was excited to see Harp talking about lipids. So we were interested, in our case, with Mars being a paleo lake, we were really interested in biomarkers that could persist for billions of years. And certain bacterial lipids like hopanoids can actually be preserved in the sedimentary rock record over billions of years. So how does this work? Bacteria present in the water column, if they die, they can be buried in the sediment where they degrade over time. And then this hopane structure will actually be preserved over billions of years, and this is because their fused polycyclic structures are quite stable. So we can then actually extract and detect these lipids in ancient sediments. So we used our Genome-Scale model to predict hopanoid and hopanoid precursor production using flux balance analysis again, and estimated a maximum of 10 to the minus 3 grams of hopanoid per kilogram of Gale Crater sediment at a single time point. And this is assuming a photic zone depth of 100 meters and assuming a single preservation event, such as settling after a mass death of 100% of the cells for one generation, and uniform mixing of the settled material within 1 meter of sediment. So I want to point out that this concentration of sedimentary hopanes is very low. It's pretty much at the limit of detection for miniaturized GCMS systems, which I believe is around 1 ppm, at least to my knowledge. But this could mean that hopanes might be detectable or at detectable levels in Gale Crater if these events reoccurred over time, for example. So in the end, this model is meant just to illustrate the potential for using genetic information encoded in life as we know it and to think more about habitable spaces on other worlds. For instance, maybe genome-scale models could be useful to inform what biomarkers could be present and at what levels, depending on nutrient ranges, and depending on the type of microbial life that could inhabit that particular environment. This type of modeling also allows you to potentially try out an idea before testing growth in the laboratory, which can be both costly in terms of time and money. We also intend to develop this modeling process further in time, so hopefully it'll be more useful at predicting and constraining scenarios of habitability. So stay tuned, and thank you.
Excellent. We'll take any questions.
Hey, it's me again. That was, again, a great talk.
Thank you.
I have a question. How have you constrained the amount of hydrogen that you use in the model?
Yeah, so I basically, what I did, everything that's going into this model is based on a flux. So what I did was utilize estimates in the ranges of partial pressure and converted that into concentration that could diffuse into the water based on Henry's law. So it's just a simple concentration flux. So that's why I'm really interested in your work, because I think that would be, great to couple it with actual geochemistry models.
Because actually hydrogen is the one I'm struggling the most. So you have that number and I have the other numbers. So we should talk.
Yeah, exactly. That's exactly what I'm thinking. Thank you.
Wonderful. Thank you so much.
Thank you.
Okay, next up we have our online presenter, Thomas Koffel.
All right. Can you guys hear me well?
Yeah.
Yep. Can you hear me? Can you hear anyone? Can you hear me?
Yes.
Okay. Sorry. Okay, good. Just making sure. So hi, everyone. I'm Thomas Koffel. I'm currently a postdoc at Michigan State University. And today I'm going to present some pretty preliminary results on this general idea of building a unifying framework for modeling the emergence and the evolution of microbial metabolisms. So before I dive in, I just want to thank my collaborators on this project, as well as my source of funding, NASA. So as an ecologist, I'm really interested in understanding this emergence of ecosystem functions, right? Like how life impacts ecosystems and how they work. And we know that it relies on microbes kind of everywhere. Like microbes help build soil fertility on Earth. They are fueling, for example, the nitrogen cycle, and also they are created the atmosphere on Earth. So if, for example, you zoom in the nitrogen cycle, it's pretty amazing to see how microbes are basically present everywhere. They fix the nitrogen, they will decompose the organic matter, they will nitrify, they will denitrify. And some of these bacteria or other microbes will be specialized, others will be more generalist. But what's amazing is the great diversity of these metabolic strategies. So biologists have started to understand really well these microbial metabolism pathways. We just had a talk on that. So it's pretty amazing the level of details we're starting to have on what happens within cells as these little chemical factories. But I would argue today, as an ecologist, I think we still maybe missing a little bit of how do we connect the two? How do we go from what happens within the cell, and how do we sort of smoothly go through these levels of organization all the way to what happens at the ecosystem level? And I would argue that what we need to do is find integrative ways of going through these levels of organizations, from individuals first to populations. So we first need to understand, how what happens within cell fuels population growth, then starts diversifying these different populations in communities, and then finally connect it to ecosystems. And I would say this is kind of one of the focus of my field, ecology. And so today I want to specifically show you that first step. How do we think about how to go using ecological theories from what happens within cells, dynamics of metabolites, to the growth of populations, using getting inspiration from ecology. And when we can do this, we have a lot of tools in ecology to start thinking about studying diversification or community ecology by just harnessing the knowledge of ecological theories. And so today, that's what I want to do, is just show you how we can use these ecological theories to scale from cell metabolisms to population growth. So let's go back to metabolic models. So we had a nice introduction on FBA with the previous talk. So I can go quickly on this. But like the standard, we have the standard understanding of what happens within a cell. So let's take this like toy model for a cell where we have a bunch of external extracellular resources, R1, R2. We have some reactions within the cell, and then these reactions, they will convert these resources into these internal metabolites. And eventually some reactions will fuel population growth. So we can formally look at this by simply saying the change through time in metabolite concentration is going to be proportional to the reaction rates. And this reaction rate, this proportionality, proportional. The coefficient in between is going to be this stoichiometric matrix here that links the two. We can rewrite this as simply, again, this product, but it's important to remember that these reaction rates, they obey some kinetics. So they are going to be functions of either the external resources or the internal metabolites, right? So the problem is not as simple as simply linear because of these kinetics in between. So now that's what happens within a cell. How do we understand how we could go to the population level? So what I find interesting as an ecologist is that, so technically, if we were to try to model population growth, you would have to follow each of these cells individually from their birth after cell division all the way to when they divide again, right? And as they do this, they are moving in this like metabolic state because they are accumulating metabolites before they can divide, right? So it's one of the properties of life. Everything needs to grow first before it can divide. And so this means that if you're looking at a population, there's going to be some heterogeneity in the state of the cells, right? And technically, if we want to do this correctly, we would have to keep track of the state of each cells in the population, maybe by following the full distribution over the metabolic state. This is obviously extremely complicated to do in practice. And so for today, we are going to ignore this heterogeneity and focus on the average metabolic state. But there are ways in ecology to actually account for this heterogeneity. But today, I will not look for it, but you can ask me questions if you're curious. So then when we do this, we find these simplified versions of models where we are keeping track of the total abundance in the population, but also the average metabolite concentration in a cell. And then we have an equation that describes the dynamics of these extracellular resources, the sort of the environment. And we can think of this as a sort of more complicated versions of the classic resource consumer models we use in ecology. Now we have a population that is more precisely described through this metabolite concentration within the cell, the population. So when you look at these equations, we find again this sort of within cell metabolic model I was just telling you about, but we have an extra term that shows up. It's called the dilution by growth, and it is a mechanistic consequence of accounting correctly for what happens when the population grows. Like metabolites get diluted as a consequence of growth, and this has been pointed out by other authors in other sort of part of the literature about metabolic models, but is, for example, not included in FBA. Now, we still have to figure out what is this expression for the kinetics of my reaction rates as a function of resource or metabolite concentrations. So you could tell me, like, let's just use the classics like Michaelis-Menten to describe this reaction. But today I want to show something a little different. Like we came up with this idea that we can represent these chemical reactions by sort of abstracting out the sort of complexity of Michaelis-Menten by instead using a minimum function. So we're saying that the reaction rate is going to be given by the concentration of the most limiting metabolite that is a substrate for that reaction. The same way that the level of water in this barrel is given by the shortest stave in the barrel. Okay. So if you, for example, take this reaction here in this toy model of a metabolic network, V1, it has two different substrates, R1 or Q4 here. And so we just write the kinetics as being this minimum function. And then if Q4 is more limiting than R1, the reaction is just directly linear proportional to Q4 with an affinity. Or if it's the other way around, R1 is limiting and then the reaction rate is proportional to R1. Okay, so it's very simple. And it has a very, very convenient property that this is a locally linear problem. So the other thing is, because we can think of each reaction as being limited by only one metabolite at a time, we can look at the whole metabolic network as looking at the combinations of each reaction, all possible possibilities of limitation. So we can count them. And then we have all the possible limitation regimes for our metabolic network that sort of qualitatively describe the functioning of the network. So you can do this here, it's only 12, but for it goes fast, I'll show you. But when we do this, like if we go back to this argument that now we have this linearity of the reaction rates, something magical happens, like for ecologists. Like suddenly we are in the realm of what we call this linearly structured population. So we have a structure for the growth of my cell populations alongside their metabolite concentration that is very familiar. We indeed recover the theory of linearly structured populations that we use in ecology in general to describe, for example, the dynamics of a butterfly laying eggs, giving birth to caterpillars, chrysalis, etc. Or, for example, the growth of like site-structured population. Like we enter the realm of like very well-known theories. And so if we want to compute the growth rate of my cell populations, we can actually just mimic what we do usually the life cycle. So we can close the life cycle. But this time it's like a metabolic life cycle. So we can actually understand the way these limitation reactions sort of feed on itself, creating this like autocatalytic loop. And we can use the theory to just find this asymptotic growth rate. So now we have this growth rate. And so I just want to show you like what we do with it. So let's take an example here with E. coli, looking at the core model and focusing on the glycolysis pathway. So I'm just going to take in glycolysis and so that's glycolysis. We're just going to assume it's a proxy for cell growth. And we have different substrates here that can enter glucose, fructose, phosphorus, and some protons. If we again think of it using this like combinatorics of the different way reactions can be limiting, you can see we have like around 2,000 possible limitation regimes. So it's like pretty complicated to just go through all possible ways this metabolic network can function, but we actually can still do it on the computer. So let's just look now at the growth rate of this metabolic network and the population along gradients of phosphorus and sugar glucose availability. So this is the result I get with this model. And so I get the growth rate of my population as a function of the resource availability on glucose and phosphate. And you can see this curve, I don't know if you can see in 3D, basically has several sort of sides. There's a first side here that corresponds to the glucose limited growth, where glucose is the only limiting resource for the cell. And if you can see, adding phosphorus doesn't really impact growth. So we are only limited by glucose here. And here it's the opposite. It's phosphate limited and glucose doesn't do anything because we already have enough of it, right? And so we get this like right angle growth surface here that is kind of a classic of theoretical ecology. It's called an essential resource. Like we need both of these resources for growth to be possible, okay? And so they sort of, and as soon as we have enough of one, we only need the other. And you can also notice we have this emergence like ceiling in growth. So this is a consequence of basically the sort of yield of the reaction combining with the dilution by growth, adding a maximal cap, where as you can see, adding glucose or phosphate doesn't really fuel growth, right? We've reached the sort of like intrinsic growth, maximal growth of the metabolic network. So this is cool because so this is one example. We get these essential resources that emerge, but we've explored glycolysis a lot more, and we find some pretty interesting interactions like between glucose and fructose. Sometimes we get inhibition when there's too much of a substrate, like it's consuming resources that could be used for something else. So it's like detrimental, etc. But I don't have time to talk about it. Another aspect that we found that was pretty interesting is we saw the emergence of alternative metabolic states. So for fixed nutritional conditions like fixed phosphate and glucose, and you look at these equilibria of this growth, balanced growth equilibria, you can be either glucose limited or intrinsically limited. And the consequence of this is that this could describe the emergence of two morphs in a clonal population that has the exact same metabolic network that grow in two different phases. So we can have basically heterogeneity in that population that emerges because of this alternative stable state. Now you're going to ask me, now what? So it is pretty cute. It's very theoretically oriented work, but what do we do with this? What do we do when we have this growth rate as a function of resources? And I would argue that in theoretical ecology, this is basically the cornerstone of everything else that can be done on top of this. So we can start to think about the microbial niches. We can quantify, the sets of conditions under which a microbe could be able to grow. We could start doing like coexistence, studying the niches. So using these niches to understand which conditions two different metabolic network would coexist or exclude each other. And we can start doing using invasion fitness to try to understand how these organisms can evolve by adding or removing reactions in the metabolic network. So this brings me to my take-home message. So basically, I hope I showed you we can use structured population theory to go from cell metabolism to population growth. We've seen the emergence of resource limitation. We have possibility for alternative metabolic states. And finally, this should open the door to doing like standard ecology and evolution to tackle broader questions. Thank you for attention.
All right, we have time for some questions.
Hi, that was a really interesting talk. Cole Mathis, Arizona State. Maybe you covered this and I just missed it, but I'm just curious if you have any ideas about the best way to validate this type of approach experimentally, or if you have thoughts about like what the easiest sort of point of contact with some observations or some experiments would be.
That's a good question. So I'm not too much in the field, so I'm not a microbiologist myself. So I don't have too many ideas how to validate it. I'm open to suggestions if you have any. Yeah.
Yeah, I'm not a microbiologist either. I was just curious. Thanks.
Okay.
I had a quick question. I saw. Can you hear me?
Yep.
Okay. So I missed something, but I saw that you were looking at phosphorus and glucose limitation. Have you thought, like, I saw that your network gets very complex. Have you started to think about adding in any other sort of limitations, nitrogen or possibly metals? That would probably get really complex.
Yeah, it would be interesting. So this was like, we wanted to first look at Ecology core model, which is bigger and has nitrogen, for example, in there. But it was a big network to get to work with our methods. We chose to focus first on glycolysis as a sort of a proxy for the whole network, which here only has these 4 limitants. But it would be interesting. So if we scaled it up to the full core model, we would have other limitations, and we could start looking at this combine multiple limitations and try to understand how they emerge and what kind of limitations do we expect. Yeah.
Awesome. Thank you.
Thank you.
Thank you very much, Thomas.
Thank you.
All right, our last speaker of this session is Cole Mathis.
Thanks.
Yeah.
All right, hi everyone. This talk will be a bit different from the other ones that just happened. So Sarah Walker was supposed to give this talk. She wasn't able to be here, and this is a project we've been working on together, so I was happy to be able to take the chance to share it with y'all. So before I get started, I just want to acknowledge everyone that's contributed to this. Obviously, Sarah and I have been working on this, and we've been working with 2 early career scientists, Sierra, who just will start at a PhD at the University of Arizona in the fall, and Pritvik, who's actually a high school student who will be attending university. It's been really great working with them. They've really taught us a lot. And I've been talking about similar ideas with Harrison Smith. We had a poster earlier in the week about some related stuff. So if you're curious about that, you can find us and we can send you the preprint or show you the poster. So I just wanted to talk today about sort of what I think are the 2 main pillars of astrobiology, the main research questions. And on the one hand, it's how do we detect life? And on the other hand, how do we make life? Or rather, how do we understand the emergence of living systems? And if you take one thing away from this talk, I hope that you take away that these 2 problems have to be solved sort of simultaneously and together, because our ability to understand the life we find and our ability to make life in the lab will depend on answering them both at the same time. So to give an example of what I mean by that, I think you can just consider a topic that comes up at AbSciCon a lot. So if you think about homochirality, if I go to a session on maybe prebiotic chemistry, I might hear talk about sort of homochirality as a necessary precondition for life. At the same time, if I go to a talk about life detection, I might hear somebody talking about homochirality as a hallmark signature of life, right? And the fact that people studying the same phenomena physical phenomena can view this in two different ways suggests that there's some ambiguity that we need to resolve in the field to understand like how it could be that a biosignature is something we're expecting to happen abiotically, right? And obviously the answer to that is we expect biosignatures to emerge over time through the process that leads to the emergence of life. But what this means is that we need to unify sort of understanding the origins of life with our approaches to detecting life. And just to give an example from the history of science about why this might be useful, you can think about particle physics in the sort of early 20th century. People were doing experiments in accelerators, and then at the same time, people were learning about sort of inflationary cosmology. And there was this realization that the early universe was very hot and dense, and it was sort of experiencing similar conditions to what was happening in the particle accelerators. And so to understand the early evolution of the universe, you could use our theories of particle physics, and then at the same time, by understanding the large-scale structure of the universe, you could place constraints on our models of particle physics. So there was this sort of feedback here that led to greater explanatory and predictive power for both fields. And what I'm hoping to convince you of is that sort of astrobiology has this large-scale cosmological question about the distribution and abundance and diversity of life in the universe. And at the same time, there's this sort of experimental paradigm about understanding the emergence of living systems. And if we can couple these, I do think we can make more effective progress and have better explanations for living phenomena. So one way to understand this is through the framework of Bayesian hypothesis testing, and specifically with life detection. So a lot of people have been talking about Bayesian life detection, especially since 2018. There was a workshop that led to 2 papers that both sort of explored this idea, and there's been a lot of really great work, some of which has been presented here subsequently. So just at a very basic level, Bayesian hypothesis testing is about how confident should I be in a hypothesis, like there's life on this planet, conditioned on some observation or some data. And if you can sort of quantify your confidence in a few things, then you can calculate this pretty easily. And those things are like, how confident were you before you had that data that there was life on the planet? And how often does that observation co-occur with data? And how many false positives do you have? So can that observation occur without life? And how often does that happen, right? And so this is just like a really simple schematic to show how these are related. This is not sort of a silver bullet for life detection. It doesn't solve all our problems, and there are some issues. And there's a really great paper that's coming out, I think, either this month or next month by David Kinney, who's a philosopher, and Chris Kempes. And they have like a really interesting critique of this approach that I think the field should take seriously. So check out that paper when it comes out. But I just want to sort of use this example to illustrate the connection between life detection and the origin of life. And first, I just want to walk through a super simple example of how this works. So let's say we're playing a game and I've got 2 coins. One of them's like a biased coin and one of them's fair. So a fair coin turns up heads 50% of the time and tails 50% of the time. And a biased coin turns up any other ratio, right? I give you a coin, you don't know which one it is. And so you decide to flip it to try to figure out which one you have, right? So you can calculate that if you know these parameters again. So maybe you know me pretty well and you think like, oh, Cole's messing with me. I'm pretty sure he gave me the biased coin. Or maybe you don't know me at all and you're like, okay, it could be either, I'm not sure. So your prior on the biased coin, this like PB, could be somewhere between 0 and 1. And then you also need to know how biased the coin is. So if it's the case that like the biased coin always shows tails, then this PT condition on B would be 1, or maybe it only shows heads, in which case it would be 0. But if you have these 2 variables, you can calculate, you know, how confident you should be that you've got the biased coin. So that's on the vertical axis, the posterior, like what's my confidence that I have the biased coin conditioned on seeing tails as a function of those 2 variables, right? So here again, you're just talking about how biased the coin is. So on the left-hand side, if the coin always shows heads and you see tails, you know you've got a fair coin. But if it shows something else or there's a different bias, then there's more ambiguity. And this really strongly depends, again, on your prior hypothesis about the chance that I gave you that coin. And this is the same data, just sort of switching the axes around here, where now I put the prior on the horizontal axis and the color is how biased the coin is. So that might seem like a sort of abstract example, but it's directly analogous to the problem of detecting life on another world based on an observation, right? So this is like one of the main questions we're interested in astrobiology is if I make some observation of a planet, how confident should I be that there's life there? And this is the exact same equation with like pretty much the same variables. I've just changed the labels from like biased and tails to life, not life, and observation. And I've changed the axes here on these plots to logarithmic scales because our uncertainty about both our prior expectation about the emergence of life, we're uncertain on logarithmic scales. We're not like, oh, 50-50 or 25% of the time. It's like, could be 1 in 10 or it could be 1 in a million or 1 in a trillion, and we don't know where it is. And so all of this is to say that like here, if we plot our false positive rate on the x-axis, which is this graph that's circled, which is how often does this observation occur when life is not present, we can sort of look at how confident we can be in life detection claims based on our prior hypothesis of life occurring in that environment, right? So if our prior hypothesis is quite low, like 1 in 10 to the 4, we need to have very, very low false positive rates to make strong life detection claims, which is sort of being at the 1 at the top of the graph there is a strong claim. And another way to sort of obviously see this is if your false positive rate is 0, then this term goes to 0 and you've got something over itself, and this term is 1, which means you don't actually need to be confident in your prior hypothesis about life being there in order to know that it's there. So another way to say that is if your biosignature has any false positives, you need a prior hypothesis about life's origin in that environment. But if it doesn't have false positives, you don't need a strong hypothesis or really any hypothesis about life's origin in that environment. So this sort of means that like life detection, there's 2 ways forward, right? One is, are there smoking gun biosignatures? And the other is, are there ways to estimate the likelihood of life's emergence given in a particular environment? So I do think unambiguous biosignatures exist. I think like an intuitive example would be directed radio transmission from another star system, right? There might be some ambiguity about decoding the signal and exactly what they're sending us, but if we got a message that was like encoding the digits of pi, we don't have to argue about the likelihood of life emerging in that star system because there's no other explanation besides an intelligent civilization, right? There's no chance that that's a false positive life detection. I think that there are other ones. This is tentative. We're trying to work out the sort of validation of this, but there's some evidence that there might be ways to use chemical biosignatures that are unambiguous. But the main point is that identifying unambiguous biosignatures requires what is and isn't possible with and without life in principle, not as a matter of scale. And that requires sort of a theory and experimental paradigms to test those measures. So what about on the other side, right? Are there ways to estimate the likelihood of life's emergence? So there was a paper in 2018 by David Kipping and his collaborator where they asked this question about what sorts of information would best constrain the prior on life. And they sort of said there's like basically 3 pieces of information we could get. We could get data from large-scale surveys of life by trying to detect it on other worlds. We could collect experimental data about the likelihood of life's emergence in the lab. Or we could put tighter constraints on life's early emergence based on the sort of phylogenetic or rock record. Large-scale surveys of life are difficult because, again, we don't have unambiguous biosignatures that work for planets outside the solar system yet, and we would need the prior to evaluate those in the first place. This review found that the data from better constraints on life's early emergence would actually be the least informative about constraining life's prior. And so that leaves this experimental paradigm where the question we now have to ask is like, what's the likelihood of life emerging in a given planetary environment? And can we make chemical simulations of that to span the space of possible biospheres in relation to the possible planetary environments? And that's a gigantic question, which we're not really approaching in a systematic way yet. And the last thing I want to end on is just that another analogy to particle physics, that's a huge question that can't be addressed in a single lab. And there are other fields in the history of science that have addressed these large-scale questions. So Supercomet-Conde is a detector that you're looking at here, and they're looking for proton decays. And this is like a giant experiment buried in Japan. And just to give you a sense of scale, that's a few people in boats working on the detectors there. And it's not detected anything yet, right? The whole point of this experiment is to look for proton decay, and it's been doing this for, I think, about a decade or more. And they've not detected one yet, but that information is still useful because it's putting ever tighter constraints on the parameters in that model. And so I think the thing I want to end on here is like, what is the analog experiment for the origin of life so that we can understand the emergence of living systems? We can make these constraints, right? And I think that it will take sort of something of the scale of the LHC or something like Supercomet-Conde to really explore the space of possible chemistries in relation to possible biospheres. Right, so I'll end there. Hopefully I didn't go over time. So the main thing, again, is just that our search for life is not independent of life's origin. We need to specifically test the hypothesis that life is on a planet. It should not be the hypothesis of last resort. We need to collect the data and information that we need to evaluate the hypothesis in its own right and not try to rule out all possible abiotic worlds because that's as big, if not bigger, than the space of possible biotic worlds. I think we can agree that there are some decisive biosignatures like directed radio transmissions. How do we find more? And if we can't, how do we make strong prior hypotheses about the emergence of life, or at least specific living processes in given planetary environments. I think for both of those, we need a deeper integration of origin of life and astrobiology, and we need these sort of large-scale experiments to try to rule out and reduce our uncertainty about the space of possibilities here. And that's it. Thanks.
Thank you so much, Cole. Do we have any questions?
Nice talk.
Thanks.
Sean Domical, Goldman, NASA Goddard Space Flight Center. I wouldn't argue with any of the conclusions with this question I'm about to ask. I think we definitely need to fund more origins of life research. My question is, short of something coming from a communicating civilization or evidence of a artificial production from something like a civilization, is there anything that you would allow as, or some combination of signals that you would allow as a decisive biosignature?
Yeah, so I think that's a really interesting question. And my answer is I don't know, but I think the way we need to go about that is understanding what is possible in principle with and without life, right? And so the reason, sort of starting from the like directed radio transmission, the reason that is unambiguous is because there's no configuration of the world that like would lead to that without life. And I think that we're in a stage where we are sort of not approaching the problem in a way where we can frame that, right? Because we know that life doesn't violate any of the laws of physics or chemistry. And so we start to like be very uncertain about what is and isn't possible. But I do think that like that's one example. There may be others. And I think what we need to do that is develop theories that help us sort of see where those might be and then figure out ways where we can test them on Earth, right? I think that's like really the challenging thing, is figuring out ways where we can build a hypothesis and also test it and validate it and then go look for it elsewhere.
Great, 300%. Thank you.
Yeah.
Hey, Cole.
Hey.
Lou Chow, NASA Goddard Space Flight Center. My question is, can you talk a little bit more about the kinds of limitations in our interpretive framework of real data that's coming from missions that are related to astrobiology, and the limitations that we have in our detection methods that can influence your probability numbers that go into the Bayesian equations? Thanks.
Yeah, I'll try. So obviously there's a few different levels, right? So what I was talking about today was sort of assuming you had perfect data, that your observation is not somehow noise, right? And that's like a theoretical sort of jump. And the reason I make that is I think when it comes to like instrumentation noise and things like that, that's like a technical question that I think specialists that already exist can sort of work out. And it's hard, like I don't want to minimize it. I think it's super challenging. But it's sort of like, I think something that we already know and understand how to solve. I think the challenge comes from like evaluating, and related to Sean's question, like how will we ever know that there are no false positives, right? It's very difficult to prove a negative. And so then the question becomes like, how can we build sort of analysis frameworks where we can really try to push the limits on what it, how do we like sort of load the dice in the favor so we can be like, okay, what's the most likely false positive? And how do we really force that? And then ask the question like, okay, could we ever actually have forced that if it wasn't for living systems? I don't know if that answers your question or if you're asking something more specific.
We can talk more after. Thanks.
Cool.
All right, thank you very much, Cole. And thank you very much to all of our speakers today. You gave wonderful talks.